Good evening, everyone. So it's a great pleasure to host today in Global IMMUNOTALK Rejane Rua and Elisa Mescolan. So Rejane Rua is a young French PI scientist. She grew up in Marseille, and at the age of 20, she integrated the very prestigious École Normale Supérieure to train as a biologist. Rejane became then interested in infectious diseases and decided to do her PhD in the Pasteur Institute studying retroviruses in the lab of Olivier Schwartz, and that was in 2010. And Rejane decided to specifically study foamy viruses, which are complex retroviruses that can cause persistent infections in animals. So for her postdoc, she joined the McGavern lab at NIH, and she became there interested in brain viral infections, so moving from the molecular and cellular scales to the scale of the tissues. And she contributed to the description during her postdocs to the description of the role of innate lymphoid cells, as well as monocyte in brain antiviral immune responses and brain neuroinflammation. Then this got her interested in some specific population of myeloid cells, which are the meningeal macrophages. And Rejane found that these cells play key roles in brain development and brain function. And she's currently studying the impact of viral infection and neuroinflammation in these processes. So this will be the main subject of her talk. So studying these cells as new player of neuroimmunology is really the main focus of her lab. She built it in 2019 at Center of Immunology Marseille-Luminy in France. And she obtained by then very important fundings to do so, such as the prestigious ERC starting grants. So Rejane has also got several important awards, including the Rising Star Award of the International Union of Immunological Societies, the EMBO Young Investigator Program Award, and the Fellow Award for Research Excellence by NIH. So Rejane, very importantly, she's also active in diverse teaching activity and scientific outreach, which we know is important to mention because it's really essential today to maintain the interest in science of the general public, as well as of politicians who decide to fund science or not. So today she will be presenting her work, and she will also leave some space at the end of her presentation to a PhD student, Elisa Mescolan, that you can see on your screen. So we're very happy to host Rejane as a speaker at Global IMMUNOTALK, and my special introductory question for her will be, Rejane, in what ways has interdisciplinary collaboration enriched your scientific perspective? Please, Rejane, go ahead.
Thank you. So thanks, first of all, thank you so much for the organization. I'm really pleased to be part of this seminar. And so, yeah, the interdisciplinary research, I think I started to fall in love with this when we started to study viruses and immunology, and now between immunology and neuroscience. And I think we go really far in each discipline, but it's really nice to now be able to see what's at the frontiers of those disciplines, and to be able to share tools, expertise, and knowledge with colleagues from another community. I think that's really enriching, and I really think that's the future of research.
Great, thank you, Rejane. I think you can share your screen and start your presentation. Thank you so much.
Perfect. So thank you again for this opportunity. I'm really excited to share today what I'm really passionate about, which is the brain borders and how we think that they're really important to control brain functions in terms of inflammation and also cognition. So as Anna-Marie mentioned, I'm a group leader at CIML in Marseille. And so we first, when we think about the nervous system and the immune system, for a long time, we thought that these two systems were kind of independent. But more and more, we realize that the nervous system can actually modulate the function of immune cells. And inversely, as we are mainly interested in the lab, the immune cells can definitely impact the nervous system. And so it's quite interesting to think about the central nervous system and its linked with the immune cells and inflammation, because indeed the central nervous system does not really tolerate excessive inflammation. First, because CNS is encased in the skull, so edema can be fatal, and adult neurogenesis is very minimal, so the neurons have to be protected because they are really hard to replace. And so when we think about the inflammation of the brain, we also think about the inflammation of the borders of the brain, including in the meninges. And we really wanted to understand whether the meninges can control the inflammation of the brain itself. And so this question started because when we think about the meninges that are between the brain parenchyma and the skull, those meninges actually contain a vast diversity of immune cell types. And this really came as a surprise, because for a long time we thought that the meninges at the brain borders were just like an inert connective tissue. But we and others realized that there's lots of immune cells at the brain borders, including meningeal macrophages, innate lymphocytes, T cells, etc. And another particularity of the meninges is that, in contrast to the parenchyma itself, where there's a strong blood-brain barrier that has been really nicely studied, the borders of the brain, which are linked with the meninges, there's no blood-brain barrier, and so the vessels are quite leaky up there. And so we think that might be one of the reasons why there's so many immune sentinels constantly scanning what's going on at the brain surface, maybe to avoid infiltration or to filter what's going in the brain. And so when this field really started to rise like a few years ago, it was really interesting to see that the meninges can actually really control inflammation of the brain in different cases. I'm going to show you little examples of what happens in case of autoimmunity and infection, but this is also true for stroke, AD, etc. And as we'll see also towards the end, the meninges can also control cognition, as it has already nicely been shown by different groups, including anatomy. So the first topic I would like to talk to you about was our initial discovery with Van der Zavich at the NIH about the role of these ILCs and the context of autoimmunity. And at the time, we really wanted to know if we can modulate immunity at the brain borders to modulate the inflammation of the brain itself. And so, as I said before, the brain borders contain a lot of immune cells, including those innate lymphocytes. And so we're quite interested in those cells. And we studied them in the context of autoimmune disease. So basically, we worked with EAE, which is somehow a mouse model for multiple sclerosis. And multiple sclerosis is a debilitating disease that affects more than 2 million people worldwide. And it's an autoimmune reaction against antigens of the brain, which leads to brain dysfunction. And during the animal models, the autoreactive T cells will invade the brain parenchyma and promote neuronal damage, which will lead to paralysis, as you can see in this picture, and death. One of the key questions was for us to try to understand, where does the disease start? Is it in the brain parenchyma itself, or the spinal cord parenchyma, or is it at the borders? And so we started to look at the meningeal immune cells, and we found that there's indeed lots of immune cells in the meninges of the spinal cord, as well as we described in the cranial meninges. And so those meninges clusters, you can see that even when the mice is not symptomatic, so the mice is going to soon develop a symptomatic disease, but for now the mice is not symptomatic, and the parenchyma is completely clear inflammation. But you can already see clusters of immune cells, so MHC2 positive APCs, CD4 positive encephalopathic cells, as well as NKp46 ILCs. And you can see that those clusters appear very early on, and then they will progress during the progression of the disease. What was interesting for us that I can go more in details later. But basically, when we get rid of these NKp46 ILCs in the meningeal space, we can actually get rid of the disease and completely abrogate part of the symptoms. So this really shows that those ILCs are important to control inflammation of the central nervous system. And what we actually found was that those ILCs that are present in the meninges, but they are actually absent in the parenchyma, so it's really something that happens in the border, they are able to perform activation and clustering of APCs and T cells, which is actually really important to license T cells, which will then enter the parenchyma and disrupt the CNS. So this was actually really interesting for us because this shows that manipulating immune cells at the borders of the CNS is actually enough to completely control the inflammation of the parenchyma itself. And so this really made us understand that the inflammation of the meninges is crucial to understand the inflammation of the brain. And after studying the ILCs, we also turned into other types of cells that are quite prominent in the meninges, which are those meningeal macrophages. And so if you just count the number of cells, the meningeal macrophages are the most abundant immune cell type in the meninges. And if you look at them under the microscope, you can see using CX3 CR1 GFP mouse, for instance, you can see that these cells are constantly scanning their environment. They are really active, always scanning what's going in and out of the blood vessels. And if you look deep down, you would see the neurons and the microglia. But here we are really at the surface of the brain, and already at homeostasis, you have this huge network. And to put it in perspective, if we look at the dural meninges, the one attached to the skull, here you have more immune cells in terms of quantity and quality than in the whole brain parenchyma. So we really have to think about the brain borders as a huge network of immune cells. And this really shocked me in a way that we always thought that the brain parenchyma was kind of devoid of most immune cell types apart from the macroglia. And we now realize that no, they are not absent, they are just located at the border of the brain, probably for evolutionary reason to try to protect brain and brain inflammation. And so if we look at those macrophages in the meninges of the brain or spinal cord, we can see that there's a huge network of cells, and you can send them with CD206. And then we have different flavors of macrophages, MHC2 negative ones and the MHC2 positive one. And when we are born, so the neonates, they start with MHC2 negative macrophages. And then as we age, we have an increase in this MHC2 positive cell population. But they all cover the whole meninges. And if you look at the transcriptomic signature, you can see that the MHC2 negative population, the one that we have neonatally, are enriched in growth of the tissue, versus the MHC2 positive one, they are more enriched in immune reaction, attraction of leukocytes, etc. So now that we understand a bit better how those macrophages behave at steady state, we wanted to understand how they would react to infection and how does infection impact their properties. So to answer this question, we use the model of LCMV meningitis, where you can basically see that the LCMV is lining the brain, so it's an infection of the CNS, and the pathology is linked with the infiltration of CD8 T cells that will kill infected cells, create edema, and this will lead to herniation and death of the mice 1 week after infection. So, and this is when we inject the virus intracranially. And so we wanted to understand how those macrophages behave when we have this intracranial strong meningitis infection. So we found first that those macrophages can actually be infected by the virus itself. And we can see that up to 30% of them get infected. And then when we look at the peak of the disease, so 6 days post-infection, you can see in green the macrophages and in pink the infiltrating CD8 T cells that come to kill the infected cells. And you can see that they contact those meningeal macrophages quite frequently. And this probably is important for them to probe macrophage infection before being able to kill those cells. And indeed, if you inject the mice with a dye that only labels dead cells, you can see that at the peak of the disease, so the mice is starting to have seizure, you can see that the macrophages are really dying, probably being killed by infiltrating CD8 T cells. And as a consequence, the proportion of macrophages at the brain borders is really decreased when the mice are infected and subjected to meningitis. And in correlation with that, there's also a huge increase of monocyte infiltration at the peak of the disease that is being recruited. And these monocytes are recruited first by interferon and then by CD8 T cells. But so they basically infiltrate the brain borders at a time where the macrophages, the meningeal macrophages, the resident one, are being killed. And so we wanted to know, okay, but how can the niche be restored after that? Are those recruited monocytes be able to engraft the niche, or do local resident cells, the 50% of resident macrophages that were present, are they able to repopulate the full niche? And so to answer this question, we could not keep our current model of LCMV because this is too toxic for the mouse. So we turned into a recombinant virus, which is less pathologic, less toxic. And in this case, the mice survive the infection and they nicely clear the virus after 15 days of infection. So when we look 30 days after infection with the recombinant virus, the mice had infection, but they completely cleared the infection. And we can ask at this time point, what is the landscape of their brain borders? What are the macrophages? Did they come back or not? And when we first look using CX3CR1 GFP mouse that were either injected with PBS 30 days before or with LCMV, honestly, just looking at the pictures, the movies, we couldn't really see broad differences in terms of density or activity of the meningeal cells. And so we wanted to know all these BAMs that were able to repopulate, so those border associated macrophages that were able to repopulate the meninges, were they coming from proliferation of resident cells or engraftment of this huge surge of monocytes that came at the peak of the disease? And to know this, we used a genetic model, CX3CR1 CRE-ER, which we call the ResiRed because all the resident cells are red. So you basically pulse the mice with tamoxifen, the whole myeloid system turns red, and the resident cells stay red. So microglia and resident macrophages will stay tomato positive, while the blood monocytes and bone marrow monocytes will become non-fluorescent because there's a turnover of the myeloid pool. And so using this technique, you can nicely see that the microglia is red because there was no really changes after infection. The blood monocytes are black. And we are surprised to see that when we look at the meningeal macrophages, to start with, a proportion of them is black because there's a little turnover of those cells. But importantly, after LCMV infection, we could see that now a huge proportion of those macrophages located at the brain borders were derived from monocytes. So this shows that this meningitis and this episode of infection and inflammation really accelerates the natural engraftment of those monocytes. And when we look at the localization of those cells, we can see that the black engrafted macrophages are present throughout the meninges. So they really seem to invade the whole tissue and to replace partially the initial resident macrophages that were dying because of the infection. But does all of it really matter? Like, what's the difference between those black engrafted macrophages and the resident initial ones? So to know that, we did RNA sequencing after PBS or LCMV infection in those macrophages. And we found that several genes were downregulated after infection, including genes linked with sensing of pathogens such as CD209, which is SINAR1. So we first validated this finding. So if we look at PBS injected mice, we can see that around 20% of the macrophages are SINR1 positive. So they are able to sense bacteria using this transmembrane receptor. But following LCMV infection, we could barely detect SINR1 positive cells. So the engrafted cells really couldn't express this receptor. And so does this matter in terms of protection of the brain? So to know that, we did a secondary challenge. So following LCMV infection, so we first use LPS as a challenge intracranially, and 6 hours later, we looked at the recruitment of neutrophils in the meninges, and we saw that this recruitment of neutrophils were dependent on SINR1. So macrophages need to sense this LPS through SINR1 to promote recruitment of neutrophils. And what was interesting was that if you look at mice either injected with PBS or LCMV memory mice, 30 days after infection, and you do the same secondary challenge using LPS, you also see this reduction of neutrophil recruitment after challenge. So this really shows that those macrophages that engrafted during the peak of the disease, they actually are better than nothing, but they are not as good as the original one in terms of immune protection, and they seem to be quite defective in several sensors of bacteria. So to conclude this part, we saw that meningitis actually drive a long-term engraftment of altered macrophages. First, the meningitis drives a partial depletion of the pool of resident macrophages. Infiltrating monocytes will come and differentiate in situ into border-associated macrophages that engraft the tissue, and they actually will engraft the niche in an interferon gamma-dependent manner. I didn't have time to show this data. And these engrafted macrophages that replace the original one are quite different, even if we don't see it right away by infrared imaging. When we perform sequencing and imaging, we can actually see that they are defective in bacterial sensing, immunomodulation, and changes in MHC2 phenotype. So it really looks like when we have naive mice, we have little monocyte engraftment over time. But upon aging and with the infection, this really gets accelerated. And so we get rid of our resident pool of BAM and start to change it for a pool of engrafted BAMs, which seem to have defective properties in terms of immune sensing. And so we made this hypothesis that basically border macrophages are quite important to nurture and help brain functions, but that inflammatory BAMs try to fight off infection and then get either altered or get replaced by engrafted BAMs. And those one can actually have lower effect on brain function. And maybe that's one of the reasons upon aging or neurodegenerative diseases, we see a deficiency of those macrophages to support proper brain function. But apart from the fact that those macrophages, those border-associated macrophages are sensitive to the infection, can they actually impact the infection and the inflammation themselves? Are they able to modulate the inflammation of the brain as ILCs were doing in the context of EA? So to know that, we first wanted to change the model, because up to now we're injected the virus directly into the brain, but this is not really physiological, because in the real life, we have pathogens circulating in the blood before getting into the brain. And so we wanted to know if those macrophages were able to sense a challenge that was given in the periphery and not just intracranial. So for that, we injected the LPS in the periphery, and we performed single-cell RNA sequencing on the meninges, so the total meningeal nucleated cells. And we could see our cluster of macrophages in both conditions, the PBS condition and the LPS condition. And we could clearly see a shift of those macrophages from the PBS to the LPS, both the MHC2 positive macrophages and the MHC2 negative macrophages. And both really respond strongly to the LPS. So they are both able to sense a challenge, even if the challenge is in the periphery. We then performed the cell-cell communication algorithm to know if those macrophages, once they've been activated, they can talk to the other cells in the meninges in the brain to maybe allow proper protection of the brain. And we found that the macrophages, especially the MH2 positive macrophages, are quite good at talking to the other cells, immune and non-immune cells of the meninges, versus the MH2 negative cells are a bit less talkative, let's say. So this really indicates that those macrophages are quite good after sensing a challenge to alert the rest of the tissue of the danger of this challenge. And so, as I said before, up to now, we are using intracranial injection of LCMV to drive fatal meningitis, but we are more interested in the peripheral model of infection, which are more not representative of what happened in reality. But when we inject the virus in the periphery, nothing really happens. So if you inject LCMV Armstrong in the periphery, after 6 days, you don't have meningitis. Basically, the mice completely clear the virus, and it looks like nothing happened. They don't even have clear symptoms. So it was, for us, quite hard to study meningitis in a mice that doesn't develop any symptom. But we didn't give up. We were thinking that maybe what's going on is that the meningeal immune response is very strong and is actually preventing neuroinvasion and preventing symptoms to happen. So what if we get rid of this meningeal immune response? Could we then detect a change in viral infection? So at the time, there was no tool to specifically deplete those border macrophages. So we used transcranial injection model of PLX, and which is a CSF1 receptor antagonist. And so CSF1 receptor is really important for macrophage survival. And when we inject this drug transcranially, so just above the bone, because the bone is quite porous, and Lisa will show you, we really have strong bone channels in the bone, we can actually get the drug to reach the meninges quite efficiently and deplete those meningeal macrophages without affecting the macroglia. And that was really an important control for us. And so we do this treatment for 1 week. So we clearly nicely reduce the number of border macrophages without touching the macroglia. And then we inject LCMV in the periphery. And so as expected, the gel mice, they completely clear the virus and are totally fine. But to our surprise, the mice that were lacking those border macrophages are completely susceptible to the virus, and they die of fatal meningitis. So it really resembles an intracranial injection of the virus. So this was for us a proof of concept that those macrophages at the borders, they are really important to block the virus from entering the brain. But how does this really happen? So what we found was that using other models, so we use the genetic model of viral infection in collaboration with Toby Lawrence, we use CD163 mice in which we inject the DT, and so we can get rid of the border macrophages. And so usually when you inject a virus into a wild-type mice, as I said before, the virus is cleared very, very efficiently, and the mice is completely asymptomatic. And you can see there's no green staining in the wild-type mice. However, if the macrophages are absent, you can start to see now LCMV leaking out of the blood vessels. And 6 days after infection, you have completely a huge network of infected cells. And at this stage, the mice will completely die of infection and inflammation. So the macrophages are really important to block viral spread and block fatal meningitis. But how do they do that, and what do they need? Do they need to sense the virus itself, or do they need to sense inflammation linked with virus infection from other cell types? So to answer these questions, we crossed CD163, so to label border macrophages with TAT1 flox mice. And so in these mice, the macrophages cannot respond to type 1 and type 2 interferon, and we can see that those mice really do not control the virus. So the virus can proliferate way more in those conditional knockout mice. And we can indeed see huge clusters of virus that we barely see usually at a very low level in the wild-type mice. So these huge clusters of infected cells are now really allowing the viral spread. And we could even see when we crossed CD163 with IFNAR flox that it was really type 1 interferon that was important to control the virus. And you can again see a huge increase of viral load in mice injected with LCMV if macrophages cannot sense type 1 interferon. So it really looks like those macrophages, they need to detect infected cells through a release of type 1 interferon to be able to fully allow their antiviral potential. And because then I told you that we have initially two kinds of macrophages. So in adult mice, we have both MHC2 positive macrophages and MHC2 negative macrophages. We wanted to know, okay, which cell type is really important and key to block viral spread and to protect us from meningitis. And so, again, at the time, there was no real tool to deplete each population separately. So we used a model that we call rejuvenation that I can detail later. But basically, this allows the system to get rid of the MHC2 positive population without changing the number of MHC2 negative macrophages. So the neonatal macrophages are not affected, but the ones that came later in life are completely disrupted. And so in this model, we injected mice, so either control mice or rejuvenated mice, and we saw that indeed the loss of these macrophages was really correlated with the increased viral load, as if those macrophages, the one that we acquire over time, are quite important to block viral spread. But how does this really work? So to answer this question, we use LCMV meningitis, and we looked at earlier time points, 2 days post LCMV infection. And as I said before, usually when you inject a mice with LCMV, even if there's no symptom and the mice is completely fine, you can already detect activation of the brain borders. So the brain borders are already starting to sense the virus and to react against the virus, including anti-inflammatory inflammatory genes and antiviral genes. What was striking for us is that in the mice that were rejuvenated, so the mice that are lacking this MHC2 population, we completely lose this response, this antiviral response, which probably suggests that these macrophages are quite important to promote a broad antiviral state in the meninges, and probably through type 1 interferon sensing. And as a correlation, if you inject younger mice that do not have a lot of MHC2 positive cells, you have a stronger viral load compared to older mice. So this really suggests that the MHC2 positive cells, the one that we acquire over time, are quite important to block viral spread, and maybe neonates are quite susceptible to infection because they are lacking this population. And so we found that when the virus is injected in the periphery and start to spread, it probably starts to release, to infect few cells, but these clusters of cells are being quenched very efficiently because type 1 interferon is being produced, activating those macrophages to promote an antiviral state. However, in the absence of those macrophages, the virus can actually spread in the meninges and in the brain, and then killing the mice. So overall, we can see that those meningeal macrophages, those border-associated macrophages, are quite good at killing viruses and allowing a broad antiviral state. There was also nice stories showing that it's also antiparasitic and antibacterial. But we're wondering, beyond those roles, beyond those antimicrobial function of those macrophages in the meninges, could we also think about what could be their homeostatic functions? And so in the almost last part of the talk, I would like to just share a few slides about how we think that those macrophages are quite important to help brain functions and neurogenesis. And we started this project because there was really a nice work showing that different cell types in the meninges are important to control brain functions. So NK cells, gamma delta, T cells, B cells, mite cells, they are all quite important to control brain functions. They actually can release cytokines that reach, after being present in the CSF, that can actually reach neurons in different areas, the prefrontal cortex, hippocampus. And these cytokines can actually modulate the activity and the firing properties of those macrophages and depend of the neurons. And so depending on which areas those cytokines act and depending on their function, this will basically trigger a different set of behavior. So this can be important for vigilance, for short-term memory, for social behavior, for tissue integrity, etc. So it's quite important to have all these immune cells at the brain borders. They're not there only to protect us from infection, but they're also there to help proper brain function. And so when we started this project, we really wanted to understand, could those macrophages at the brain borders also help the brain and not only lymphocytes? And so macrophages could help the brain in so many ways. It could block peripheral toxins. It could, as been shown by Yannick, it could influence the flux of CSF. As was shown recently by Dan Mattis, could influence the other immune cells. Could these macrophages could release growth factor or impact the metabolism, the microenvironment with the vessels, the oxidation, could modulate macrophage network activation. And this could indirectly affect neuroprogenitors, proliferation, and function of the brain. So this topic was quite diverse. So we set up a bunch of techniques to try to understand this process, like could macrophages really help brain functions, focusing on the hippocampus. So we did different work with RNA, bulk RNA sequencing, of the meninges of the hippocampus, single cell RNA sequencing, and protein work with the secretum and mass spec of the CSF. We did different algorithms to try to understand how those macrophages interact with the neurons using different types of cell chat, NicheNet, SCCOM. We also looked at metabolite interaction, etc., etc. And so what we found was actually that those macrophages are quite important to help brain function, and are really important to promote brain activity, both in terms of neuronal activation and in terms of neurogenesis in the hippocampus. And we found factors, I don't have time to describe everything here, but we found factors that were important to promote these functions, both neurotrophic functions and neuroactive functions, from the macrophages to the hippocampus. And in the last part of the talk, I will let Elisa speak, but basically we're going to show you that the meningeal immunity is interesting, but it's even more interesting to understand where does this meningeal immunity comes from. And we really wanted to know if we can harness the skull to brain axis to boost immunosurveillance.
Thank you, Rejane. Okay. So I'm Elisa, I'm a PhD student with Rejane, and I'm really interested in the interface between the dura mater and the skull, and in particular those bone channels that are connecting the hematopoietic niche you found in the skull to the meninges. And those channels have been really recently described in 2018. And since studies have shown that cells can migrate from the marrow to the meninges, and it is kind of an emergency pool of immune cells sitting on the brain in case of an injury or damage. We know also that the cerebrospinal fluid can flow in those channels and can reach the marrow and influence hematopoiesis. So a lot of studies now are working on those channels, studying the role in different pathologies, meningitis, stroke, EAE, a lot of interesting things. But for now, we don't have any tools to modulate those bone channels. And this is what I'm trying to do. So first, I try to find a way to visualize those channels. So I've done biphoton intravertebral imaging. And we can see in the developing skull that you have plenty of those bone channels that are going through the skull. So it is really fascinating to see that there are so many of them during development. So what I first try to do to see with the system we are working with is to deplete the myeloid cells. And when we do a depletion of those myeloid cells, we can see with bulk RNA sequencing that we have a lot of downregulated genes, including some that are linked with bone resorption, bone remodeling, and osteoclast differentiation. So something is happening with the skull. So first, I've tried to target the osteoclast, because the osteoclast, there are macrophages that are specialized in degrading the bone. So we were thinking that maybe they are important in digging those bone channels. So I've first started by inhibiting those osteoclasts, and we can see that we have a treatment that is quite efficient, as we see less trap staining, which is basically the activity of this osteoclast. And when we do so, we see decrease in the formation of those channels. As we can see, they are smaller. So this was quite interesting for us. And so the next step was to do the opposite, try to increase the activity of the osteoclast. And when we do so, we can see an increase in the size of the channels. So now we have tools to either increase or decrease the channels. And we want to see, does that actually impact neuroinflammation? So to test this question, we had to set up a model where we could see the immune cells coming from the skull to the meninges without having influx from the blood circulation. So to test that, we actually perfuse the mouse and take the skull to incubate it ex vivo in an inflammatory medium. And then after the incubation, we peel the meninges off and look at the infiltration of immune cells. And what I was able to see is that in an inflammatory condition with some LPS in the medium, we have more infiltration of neutrophils in the meninges compared to the PBS condition. And those neutrophils can only come from the skull marrow. So this seems to be working well. So we applied it to mice that were previously treated, either to inhibit the osteoclasts or activate them. And when we do the inhibition, we can see that we have less infiltration of neutrophils. And when we activate the osteoclast, so we have bigger bone channels, we have more infiltration of neutrophils. So this is, for us, really interesting results. And so we can see that for the first time, by modulating the size of the channels, we can actually modulate the immune infiltration to the meninges. And so for now, it's only ex vivo, but we are also validating it in vivo. So that's it for me. Thank you for your attention. And I'm giving the floor back to Rejane.
Thank you, Elisa. So yeah, so basically, I think we showed you that we really think that the brain borders are quite important to control brain inflammation, as I showed with the LCME model and the brain function as well, as I showed during promotion of neurogenesis. Elisa also showed you that these immune cells actually, where they come from really matters. And I didn't have them to show, but the lab also studies what's going on during the development, and can these brain borders control not only what's going on in the adult stage, but also the formation and the development of different brain structures, including hippocampus and cerebellum. And one of the aspects of that is that we are trying to understand whether what happens in the normal development, can it be altered during inflammation, and could this be the routine cause of neurodevelopmental diseases? Could this be actually disease of the borders? So I would be really happy to chat about that later, or maybe after the talk. But I'm really happy to have shared these stories with you. I would love to thank all the team members of the lab, the previous members, the institute, and all the current collaborators. I didn't have time to list the previous collaborators, but I thank you also. And we're happy to take questions on the chat. Thank you. Thank you so much.
So thank you, Rejane, for this wonderful talk. Thank you, Elisa, for your presentation. For the questions, actually, the questions will be made directly to Rejane via Twitter, as indicated, via X, now I should say, as indicated in the slide that is being shown to you. So do not hesitate to ask all your questions and comment on these beautiful stories on neuroimmunology and these beautiful images and movies as well. I will now end just by announcing the seminar, the Global IMMUNOTALK seminar of next week, which will be given by Will Wood. So Will Wood works in the University of Edinburgh. He's an amazing fly immunologist, and he will be talking about clearance of apoptotic cells by macrophages in the fly and how important this is for fly development and survival. So thank you to all of you, and see you next week. Bye-bye.
Thank you. Bye-bye.